Grass. There's a lot of it on this planet. Excluding the Arctic, grass covers almost 41% of our planet's surface. So when we think of agriculture, why is it that we think of food crops instead of grass? In fact, turfgrass is an integral and very important part of agriculture, as well as our society. Turfgrass in particular receives an intense amount of care and attention. In 2002 alone, care of turfgrass accounted for almost a $58 billion impact in the U.S. Cool season turfgrass is a major crop in the northeastern United States and is grown in home lawns, recreational parks, athletic fields, cemeteries, and golf courses. Can you guess where turfgrass receives the most care? The U.S. golf economy generated $68.8 billion in 2011, almost doubling the economic impact of spectator sports, which includes baseball, basketball, football, and hockey combined. Golf is a significant portion of the U.S. economy and is important not only as a sport, but as an industry. A major part of golf course management is disease management. Disease management is the prevention of turfgrass disease from pests, such as insects, worms, bacteria, and fungi. Sclerotinia homoeocarpa is the fungus that causes dollar spot, the most economically important of these diseases. The most effective way to control dollar spot is to use chemicals called fungicides, which kills fungi. However, repeated use of fungicides has led to many cases of resistance, similar to antibiotic resistance. Fungicide resistance is when a particular population of fungi becomes immune to fungicides. Particularly troublesome is dollar spot's multi-drug resistance, meaning a variety of different fungicides are ineffective in killing it. Populations of multi-drug resistant dollar spot are especially difficult to control, and can easily cost golf course companies thousands of dollars. Researchers in turfgrass pathology aim to identify new drugs to use against dollar spot, as well as develop integrated pest management strategies to minimize inefficient fungicide use. To discover the underlying reasons that some populations are becoming resistant, researchers take a look at DNA to compare differences between populations. If a consistent gene is mutated only in resistant populations, then it's reasonable to say that that gene might play a role in fungicide resistance. To find these mutations in dollar spot, my lab sequenced all of the DNA of two individuals, one which is sensitive to fungicides and one which displays resistance. Researchers look at DNA in terms of nucleotides, or the individual molecules that make up all of the genetic code. Luckily, there's only 4 molecules called A, T, C, and G. Dollar spot's DNA code is relatively small, only 45 million nucleotides. Sequencers are a great new technology that can tell you the order of nucleotides that make up a genetic code. The problem being that sequencers only give you sequences in a string of 100 nucleotides, not all 45 million in one string. Assembling all of the DNA in the correct order becomes a heavy task, especially when given millions of 100 nucleotide strings. It's similar to building a puzzle. My research was to solve this puzzle and put together these strings of nucleotides to accurately represent what the genetic code of the two isolates are. With some knowledge and a very powerful computer, I was able to complete these 2 sequences. In the future, my lab and I will analyze these sequences to better understand how a resistance is obtained and how we can fight resistant populations of dollar spot. Feel free to contact me with questions or check out our lab website. Also, I'll be presenting at the Undergraduate Research Conference on April 22nd, so be sure to swing by. Thanks for listening, and remember, stay green out there.